Thank you. Um, can you see the slides okay? Like, is it a full screen slideshow?
Yes, it's perfect.
Okay, that sounds great. Shall I start now?
Yes, please.
Thank you very much for the kind invitation to discuss our work on suspect screening methodology that helps us to uncover hidden chemicals in pregnant women. So this is work that I do in collaboration with a lot of uh, scientists at UCSF as well as UC Berkeley, which I will mention in the end. So I'm Aolin Wang. I'm from UCSF program on reproductive health and the environment. Today, I would like to take you all through the journey of new discovery of previously unidentified or undocumented chemicals in pregnant women. So here are the things that I would like to cover today. So first, why do we want to screen for more chemicals? And then how do we do that? And in this project, we take the approach of suspect screening, and I will briefly talk about how how did that work, and also present the result on identification of novel chemicals. So here I would use the word novel to mean actually previously undocumented or have not been biomonitored chemicals. And then we can have some discussions. So this is a graphic showing you over the past 50 decades, the U.S. chemical production actually increased more than 15-fold. So that's quite a lot. So each year, more than 30,000 pounds of industrial chemicals has been produced for each person in the U.S. That's actually more than 15 times of the amount of food that one can eat annually. So that's a lot of chemicals that's out there. But unfortunately, unlike the pharmaceutical drugs, most of these chemicals in the marketplace have not been tested for their safety. So in our previous study, where we used the data from the ENHANCE, and what we found is that virtually all the pregnant women in the United States have at least 43 chemicals in their body while pregnant. And these chemicals can get into pregnant women's body through like contaminant air, water, or like food. And also a large portion is also coming from consumer products such as cosmetics. So currently, the U.S. National Health and Nutrition Examination Survey, or U.S. ENHANCE study, has been biomonitored for about 350 chemicals, but that only makes a small fraction of the over 8,000 high-use chemicals that's being produced and used in daily products. So it's really the tip of the iceberg that we are monitoring. So there are like a gigantic amount of chemicals that what we call unknowns. We don't have any information on. And those unknowns can further divide into known unknowns or the unknown unknowns. So here, the known unknowns are what we call the suspects because we know what those chemicals are, but we just don't have any information on them, or like we haven't done thorough research on them. But the truly unknown is the chemicals that we may not even know what they are. And today, we are going to take a look at a new technology that's called high resolution mass spectrometry, or in short, HRMS, to kind of address this issue. And in the hope to use a blood sample or some kind of biological samples to screen for more chemicals at a time. So before I go into details of our study, I think for the audience, it would be helpful for me to just briefly introduce what the high-resolution mass spectrometry technique is and how the data have been generated. So if you recall, like chemistry class, one chemical have two really important or distinct features. One is its mass, sometimes can be captured by its formula, and the other piece is structure. So based on these two features, the HRMS system has two important parts. One is a chromatography system, which can be liquid or gas, depending on the type of chemicals that we are examining. And the other part is called a mass spectrometer, which is used to detect the chemical mass. So in that sense, for the LC system, it helps us to separate chemicals based on its structure, and then the mass spectrometer can detect mass. So combining the two, we are able to get a fingerprint of the chemicals that we want to examine. So the data output is actually a series of what we call the peaks. So as you can see in this graphic, there are several peaks on at the different retention time. So the x-axis here is the time where the LC system eludes the chemical. So it's related to the chemical structure. And then on the y-axis here is denoting the M to C value, which is basically is equivalent to the mass. So in this graphic, we can see we have two important components here. And the third component here is the height or the intensity of the peak. Sometimes we use peak area to denote this value. It's a relative concentration metric in the sense that we can use this value to compare the relative intensity or relative concentration of the same chemical across different samples. So the output is what we call the signature of chemicals or features. They are basically the peaks that's denoted by a unique combination of the mass and the retention time, or RT. And in this study that I'm going to talk about, we are in close collaboration with Dr. Roy Girona's lab at UCSF, who is our expert on analytic chemistry. And the machine that we are using is this Angelin's LC quadruple time-of-flight mass spectrometry. The left-hand big part is the LC system, and then it gets charged, like the chemical gets charged and then collide with some energy, and then the fragments will fly through the very large tube here you see on the right-hand side. So that's why it's called time of flight, because the mass is determined by the time that the fragment or the chemical fly through the tube. So it's very interesting. So here, as I mentioned before, in this study, the approach that we are taking is called suspect screening approach. So this approach is combining non-targeted data acquisition with targeted data analysis. So what do I mean by that? So for non-targeted data acquisition, we are basically capturing all the possible chemical features in the samples. However, there are just too many of those features to sort through given like the limited time and human resources that we have. So we further developed what we call a suspect database. This database covers a group of chemicals that's of interest to us, and it will serve as the roadmap as we try to sort through all the chemical features that we identify through the LC-Q-TOF system. In particular, in this study, we focused on a group of chemicals called environmental organic acids, or EOAs in short. And the goal is to identify novel EOA exposures in serum during pregnancy. And you may wonder, what do we mean by the environmental organic acids? So it is the environmental organic compounds with at least one ionizable proton. This is a very technical definition, but in general, it covers a lot of the widely used chemicals in consumer products. And also, this group of chemicals have the potential to disrupt our endocrine system because they include a lot of chemicals such as phenols like bisphenol A or triclosan. And also, because of its chemical structure that has at least one ionizable proton, it has a higher rate of analytical detection under our current method. So that's why we decide to focus on this group of chemicals in our this study, and we currently we actually expand this to a much larger list. And the suspect screening analysis consists of 4 major steps. One, as we mentioned before, is called non-targeted chemical analysis, where we gather all the possible chemical features. And the second one is the data processing part, where it includes a lot of programming as well as data quality checks. And the third part is, from all these features that we detected, how are we going to prioritize or select certain chemicals that we want to further pursue in terms of confirmation. And the final step is we purchase the synthetic standard for the selected chemicals, and we confirm whether that chemical is present or not present in the current serum sample. So first, for the chemical analysis, we actually have an ongoing cohort of pregnant women that has that Tracy Woodruff is the PI on recruiting women from the 2 big medical centers at San Francisco. One is the UCSF Medical Center, and the other is the UCSF Zuckerberg San Francisco General Hospital. From that, we recruited pregnant women during 2nd trimester. And for this analysis, we actually use serum samples from 75 women. And for this 75 women, their mean age is about 33 years, and most of them delivered term babies, and about half of them have high socioeconomic status. And you can also see on the diagram or the pie chart on the right-hand side that we have a very diverse population here, although the majority of them are non-Hispanic white or Latinas, but we also have a few non-Hispanic Asian and African Americans. Using those serum samples that collected from pregnant women at delivery, we run through the LC-Q-TOF MS machine, and then we obtain the chromatogram features. So if you can recall from the previous slide that in this chromatogram peaks, we can get the chemical mass, retention time, as well as relative concentration. And the second big step is data processing. And there are several little steps within this gigantic step. The first is, as I mentioned, we use a suspect screening approach, which we will create a roadmap of library of chemicals. So here we actually create a library of about 700 compounds with 590 unique chemical formulas in our database. And the chemicals are from 8 chemical classes, including a lot of pesticides or pesticide metabolites, as well as phenols and some pro and polyfluoroalkyl substances, as well as phenolic or phthalates. From that database, we actually obtain chemical formula and exact mass for a specific chemical. And from a previous step where we got the peaks detected by the LC-Q-TOF-MS and obtained the retention time and accurate mass. So then we merge the two together by its formula. So after that step, what we get is a group of mass matches that uniquely linked using their chemical formula. From all the features that we detected, we got about 5,300 mass matches, which correspond to 248 unique chemical formulas in these 75 participants. Later, because of quality checks, the chemical experts on our team perform a total iron chromatogram peak review, where he and his team subtract features that appears in the blanks or features that appear to have very poor peak shapes. And after that, from the 4,610 mass matches, I perform isomer distinction. So here, isomers are the compounds that have the same chemical formula but differ in their chemical structure. So they differ in their retention time. So based on their retention time, I group the features that have very close retention time and the same chemical mass to be the same group of isomeric compounds. And from this grouped isomeric compounds, that's what we call a suspect EOA. A suspect EOA means a group of chemical features or chemical peaks or mass matches that have the same exact formula and similar retention time. And you can think of them as a unique chemical feature. So recall this graphic where we have x-axis to be the retention time and y-axis to be the mass. Here is a similar graph showing you all the features that we capture after the data processing part, that is after matching to our database. So you can see along in about 10 minutes of the whole elution period of time, there are actually 455 suspect EOAs that we have detected. And according to the color, which is corresponds to the chemical classes, we can see that we found a lot of phenolic compounds, and also we have not detected any suspect OHBDEs or OHPCBs. And the size of the bubble denotes the detection frequency among the 75 women. So that means the larger the bubble, the higher detection frequency this particular EOA has. So as I mentioned, we detect a bunch of predicted pesticide metabolites, phenols, or phthalates metabolites. And this is a graphic breakdown by participants. So the maximum number of suspect EOAs detected in one participant is 74, and the mean is 57, with the minimum to be 32. And the majority of them are, as previously mentioned, phenols. Regarding the isomeric compounds, we found out that a lot of the isomers that we have detected are actually from phenols or silane metabolites. And in this graphic, when we see two bubbles or more than two bubbles within the same line, horizontal line, it actually represents isomeric compounds because they have the same chemical mass but different retention time. But unfortunately, although we have detected a lot of the suspect EOAs, the majority of them actually of very low detection frequency, as you can see on the left-hand side here in this histogram. Only 12 suspect EOAs have detection frequency over 80%, and about 19 suspect EOAs have detection frequency over 20%. So among those that have a higher detection frequency, the question would be which ones do we choose for validation? So that comes to the next part where I perform additional association analysis with demographic variables, including race, ethnicity, education, and income, to decide to determine if there's any demographic differences in terms of the relative concentration by all these variables. And using the Fisher exact test or the Cruz-Gonzalez ransom test with an alpha set to be 0.1, we found that 13 suspect EOAs that shows any demographic differences. So next, apart from using information that we can obtain from the current sample or the current cohort, we also want to get some external information on the chemicals in our database to further help us prioritize which chemicals to select to do the confirmation. The reason that we want to actually do this prioritization step is because we have limited resources to purchase this synthetic standard, and then actually needs to run with the sample again through the same system to confirm its identity. So it is a little bit time consuming, so that's why we want to further select the chemicals that's of most interest to us to do the validation or confirmation step. So for the external information, we want to know whether it has been biomonitored by the NHANES or not, or the Biomonitor California program. And the second thing is we want to gather some usage information and toxicity information through all the publicly available sources. And what do I mean actually by getting the external information here is, so for example, for the 12 suspect EOAs that have over 80% detection frequency, we further match it to our EOA database. It actually matched to 21 unique compounds based on their formula. And you can see some of this. Sorry, I don't know why the slides just keep going, so you may see like little things pop up from time to time. So for example, on the second row here, because it can be an isomeric compound, so there are more than one chemical candidate that map to the same chemical formula here. And based on the chemical candidate names, we gather information about their biomonitoring status, as well as some production volume information. So from what you can see on the right-hand side of this table, 2/3 of the matched chemicals have actually not been biomonitored by either NHANES nor the California Biomonitoring Program before. So indeed, we may be able to find some novel chemicals here. And the last step is we must have a synthetic standard available on the market so that we can actually purchase and verify. So just quickly summarize the selection process is that we gather information both from the sample as well as from outside sources and selected 20 compounds in total for further validation by analyzing the synthetic standards. So it took the lab a while to actually sort through all of them because it's actually an iterative process. It's not like we select 20 at once. It's more like we select 5 or 10 first and to see how many we get, and then do that on an iterative process. And we are very happy that we finally confirm a bunch of them. So eventually we confirmed 6 chemicals that's listed in this slide. So you can see I ordered them by their detection frequency in the sample, except for the later one. But 2 of them actually have a production volume to be 10 to 15 million pounds per year, which is quite high, and the data is actually obtained from the 2016 chemical data reporting. And you may wonder, what are these chemicals and what do they do? And we look at the chemical and product category database from EPA and gathered some usage information for each of them. As you can see, not all of the chemicals have been included in the database. So, for example, the second one, we cannot obtain absolutely any information from the literature or the existing databases. But for the other chemicals, you can see they are widely used in toys, personal care products, cosmetics, pesticides, food additives, which we can all get into contact in our daily life. And even more concerning is that some of this actually have been shown to have some health concern. So, for example, the 2,4-dinitrophenol has been found to cause cataract formation. This is very interesting because it was once used as a diet control pill in the 1970s, I believe. And then the FDA banned it because it's been found to forming cataract in young adults. And also it is suspect to have some to cause some genetic defects in newborn babies and potentially could damage fertility and the fetus. And the pyrocortical chemical has been classified as the group 2B possible human carcinogen. But for the other chemicals, there have not been much data on the toxicity or health information. Although we detected some novel compounds, there are some limitations of this current study. So the major limitation is that in this study, although we deliberately focus on the group of environmental organic acids for their better detection rate, we also restrict our chemical space to be a very limited space of 700 chemicals. As you may know, there are just thousands of chemicals out there, so we are only capturing a small fraction of that chemical space. And also with this technology, it's known to have relatively low sensitivity for chemicals with low concentration or have short biological half-lives. So we may not capture all the chemicals that we want to capture or of interest to us. So that's why in our ongoing work, we actually expand this approach or this analysis on several aspects. The first is that we run all the samples in both positive and negative ionization modes. While in this study, we only use negative modes in order to incorporate the chemical structure. But with the positive and negative mode combined, we are able to capture a larger chemical space. And also, we include a lot of the chemicals that's in the 8,000 high-use industrial chemical list. So we expanded our chemical database. And for the next step, we have also developed targeted methods for quantifying the serum levels of the 6 confirmed chemicals that we found before in 2,000 maternal serum samples. But because the chemical structure differs so much, the analytical chemist cannot make all of them into one panel. So eventually, we end up a panel of 3 of the identified and pick additional 3 to make the 6 chemicals panel. And also, some other ongoing work is we are trying to extend this analysis to identify undocumented chemicals in paired maternal core serum samples, which will be very interesting because it will be the first time where we can actually screen for more chemicals in the core blood, which can give us a better idea of the chemical exposures in the fetus environment. Just want to recap what we have talked about before, because I know it's a little technical, so I would love to just briefly summarize what we talked about. For the suspect screening approach, it actually consists of 2 major pieces. One is the chemical analysis using human serum samples, and the other is to build our roadmap, which is the suspect chemical database. After that, we overlay the 2 and get some suspect features. And after some statistical analysis to prioritize which candidate chemicals that we want to select and purchase the synthetic standard, we further do the confirmation steps and confirm whether the chemical identified is actually present or not in the blood sample. It is, we think that suspect screening is a complementary and valuable supplement to the existing targeted biomonitoring methods, in that through this 4 steps approach, we can efficiently identify and prioritize novel chemicals for either further biomonitoring studies or for toxicity or epidemiologic studies to study their health effects. And finally, we hope to bring more of the unknown chemicals into light. With that, I would like to thank Dr. Tracy Woodruff and Dr. Marina Sirota, who are my mentors during the program. And also, I would like to acknowledge all my co-authors, especially Dr. Roy Girona, whose lab performed all the chemical analysis, and also all the participants and people that have given valuable input to this work. And also US EPA as well as NIEHS, March of Dimes, and the Preterm Birth Initiative for their funding support. And with the last note, I would like to point out that if it still feels a little too technical for you, I suggest that you check out my blog in both English, or if you happen to know Chinese, you can also read them in Chinese. It's preheussf.blog. I have written a blog post on our study in layman terms, so hopefully it can help people to understand this technology and the findings better. And also, I would like to share with you that this study, together with a statement on food additives in Child's Health published by the American Academy of Pediatrics has been covered by the New York Times. So if you are interested, you can do a search for the New York Times article as well. And I'm happy to take any questions from the audience, or if you have any concern or any questions, you can also feel free to email me. Thank you very much.
Dr. Wang, thank you so much for such an interesting and great study. And it's actually very interesting. And at the other side, it's so frightening that we are exposed to so many known and unknown unknowns that we really don't know what they are, and we are exposed to them every day, and we don't know what are they doing with our body. Okay, so any questions from the audience?
This is Jessica Kastner. Thank you so much for this excellent presentation. Can you comment based on your experience or your thoughts about the difference between using serum or plasma samples with this method?
Regarding serum versus plasma, I actually don't know the technical aspect of why one would love to use serum over plasma, but from the labs that I have been working with, or the labs that I heard of, if you want to do like suspect screening or sometimes metabolomic studies, serum is a more commonly used matrix compared to plasma, but I just don't know. But I'm happy to like forward your question to our analytical chemist who may have a better idea of why that is. And another point is actually there are some people who are interested in this work also ask about whether we can do this in urine samples, because as you can imagine, serum is very valuable, and actually a lot of the cohorts don't have that much serum, but we can easily collect urine. But the one extra challenge with the urine samples is that for a lot of the chemicals, we actually don't have much information about how they metabolize. And in urine samples, we usually detect their metabolites. And depending on like how the metabolic pathways goes and what kind of biomarker is actually reliable or available, urine is more complex in that sense. So apart from one study, I haven't seen any other studies that's doing either non-targeted or suspect screening work using urine. That's just like an extra thing.
No, that's excellent information, because a lot of our nurse scientists or affiliate scientists, they're looking at the matrix. They're facing a clinical problem, and they're looking at the matrix of, what sample can I take? How do I process it to even get it to the lab, at your point, which is where, our disciplines and our work would intersect. And so that's incredible. And even, your thoughts on saliva as a potential. I'd love it if you could forward my question, and I'm sorry that I asked such a difficult one, but your answer was eloquent and to the point. So I'll open it to another question. Thank you.
Thank you.
So, Aolin, I have a question. Did you take a survey in regard to possible sources of these chemicals from this population? For example, their occupation, are they exposed to any occupational exposures, or do they smoke, do they work in the farm or something like that?
Yes, I may not have mentioned this earlier. We do have a very extensive questionnaire that's given out to women during their second trimester. we ask about their occupation and their consumer product use. For example, like the daily use of the very common consumer products such as household cleaning products or cosmetics, lotions, etc. And we also ask them their their food consumption like preference, or for example, like do you eat out, like how many times a week? That question is mostly to get at the exposure to either the perfluorinated compounds or the PBDEs, but that also can be additional information that we can probe. And because this study is in collaboration with Dr. Rachel Morello-Frosch, who is an expert in psychosocial stressors, so we have also an extensive questionnaire asking them about their social or psychosocial stressors. So from home and work, I guess we may be able to further pin down where these chemicals are maybe associated with or are coming from based on their occupation or their consumer product usage. Actually, I'm doing a follow-up study trying to link all these suspect screening products to suspect screening chemical features to the consumer products used for among these women. But I haven't found any like super interesting results yet.
Great, thank you so much. That's very helpful.
Thank you for your question.
Any other questions?
Hi, this is Luce Huntington Moskis. I have a question. I very much enjoyed the presentation, Dr. Wang, and I noticed that, as you mentioned, Dr. Morello-Frosch is part of your team and a collaborator, and I'm familiar with her work because of her report back work that she does. I'm just interested if your team has had any discussions on that. I'm sure that the idea of report back with these unknown unknowns is very ethically concerning, but I'm just curious as a research team how you've at all discussed that point with this particular study.
Wow. First, thank you very much for the question. It is a really great point because we have some discussion among our teams about whether or not we should disseminate the suspect findings, and if so, like how. Because for most of the report back work before, we actually report the exact chemical concentration to the participants. But in this study, the situation is a little bit tricky because from this study alone, we cannot get the absolute concentration of each chemical. And not to mention that apart from those 6 confirmed chemicals, we actually cannot be 100% sure what other the suspect chemicals actually are, although they have been matched to a particular chemical name. But until we further confirm it, we cannot say that that chemical feature is the chemical that it's linked to. So there is, it is not 100% clear if how are we going to disseminate the result with so many like uncertainties to like a general pregnant women. I think it may not be very useful for them because at first, if we want to disseminate, let's say the 6 confirmed chemicals, then we may be able to say the concentration, but without any context. because first, we don't have much information on their toxicity or any of the epidemiologic studies out there, and also biomonitoring studies, because in this current study, we strictly restrict to the chemicals that have not been biomonitored before. So that means we don't actually know across the across the U.S., what is the concentration distribution look like. So we don't. It's very hard to interpret what that value actually means. And apart from that confirmed chemicals, for the rest of the chemicals, because we are even not sure their chemical identity, so it will be very challenging to actually present it in a correct way or a easily comprehensible way, I would say. So at this moment, although we have talked about that, we don't have any plans to actually disseminate this result to the participants. I think it's more for the research community to see what other chemicals that we can do more research on and try to get a better understanding of the previously unknowns.
Thanks for your response. I just find it very exciting that you've had these kind of conversations, even if the end result is we need to withhold that for the moment. I think it's just wonderful that your research team has kind of grappled with some of these issues.
Yeah, and Dr. Woodruff actually would love to make the data publicly available. So we are actually working to make like all the QTOF data available to the research community. So if other researchers want to do a similar approach or like do the non-targeted approach, then the resource is shared. So in that sense, we hope to kind of move the field forward like faster so that we get a better understanding of all the chemicals out there and take better action at it.
That is great. Any other questions? So it seems like there is no other question. Jessica, do you have any question?
I put a few in the chat just in case others wanted to ask. I do have 2 additional questions while we still have time with you. This is just fascinating. The first is, and if other people want to jump in, please don't let me monopolize the time, but while we have your expertise, are there other vulnerable populations this suspect method has been used in your literature review? And if so, were there any surprising findings? So, you know, besides the pregnancy course, were there other populations that this has been used in?
To my knowledge, up until we published the paper, which is like July last year, I haven't seen any suspect screening approach that's used specifically in human serum samples. So we are very happy that we are the first in that sense. But I haven't do like a updated literature search after that. But I know there have been a lot of efforts trying to do untargeted metabolomics using the serum samples, but that's like for another different focus. But for environmental chemicals perspective, I think this is the first study. But in terms of other vulnerable populations, I guess the other one that we are actually currently doing is the cord serum, which is like like complementary to the maternal serum, which gives us a better sense of the fetal environment. So we are actually hoping to get analysis done pretty soon and publish some preliminary findings.
That's wonderful. That answers my question. And the next is more of a comment or a recommendation. I wonder, of the 6 chemicals that you found, if using the comparative toxicogenomic database, it's at NIEHS has put out a few papers in regards to it, and we can certainly send you the link, but I wonder if putting those 6 chemicals in would help identify the link to diseases or sources as well.
Yeah, that's a very great suggestion. I actually come across this database at the ISEE meeting. So I think I'm going to take a look at the database. It seems to be a very extensive and very useful database in the sense it links like chemicals with the underlying genetic features as well as the disease signature. So it completes the linkage, so you can find very useful information there. But thanks for the point. Yeah.
Absolutely. And thank you again. What a great presentation.
Thank you. And if you don't mind, Jessica, can you maybe email me so that I get your email and I can kind of ask the question about like serum versus plasma and get back to you?
Absolutely.
Thank you.
And Dr. Wang, do you have any plan to follow up the amount of these chemicals in these babies and see if they are passing through placenta or not?
Oh, do you mean, do we have any follow-up studies for the placenta?
The placenta, and also if you follow up those children, you know, up to age 1 or 2 or whatever, you may find, you know, the amount of the concentration of these chemicals in children's body of these pregnant women.
Right. Yes, very great thing is that Dr. Tracy Woodruff and Dr. Morello Frosch, they have been involved in this ECHO study, which they got additional funding to follow up the pregnant women and their babies up to, I think it's age 4. So as the cohort like ages, we actually can get some additional outcome information from the child. For example, their neurodevelopment outcomes or their general health status or their weight, something like that. And because the data on the chemical side has already been generated, so we can actually do some new analysis to look at whether some of the chemical features are like show up more or at a higher level among people, for example, like among child with say impaired neurodevelopment outcomes, something like that. It's a plan. We haven't gotten enough data to do so yet, but it's in the pipeline. Thanks
Wonderful.
for bringing that up. Yeah.
Thank you so much. So when the data is out for the follow-up study, just please let us know. We shall bring you back and then you'll give us another presentation on the new data. We will be excited to hear that.
Thank you. Yeah, I will.
Yeah, appreciate it, Dr. Wang. You have a wonderful day.
Thank you. Same to you.
Yeah.
Bye-bye.
Bye-bye.